Hi, I'm Veronika from the Genesis team. We are pleased to present our project in the proposal category at the Global Open Genetic Engineering Competition. As you can see, our team proposes the construction of a bacterial biosensor of interferon gamma based on Escherichia coli cells. First, I would like you to get to know our team. We are a group of 12 students, 6 of which are currently working on their master's thesis, but when the project started, 9 of us were undergraduate students. We all belong to the Synthetic Biology Genesis Students' Society, affiliated with the Department of Biology, University of Warsaw in Poland. Our society was founded in 2008 and was recently reactivated in 2021. Genesis project aims to find an affordable, accessible, and easy-to-use alternative to present latent tuberculosis infection tests. My fellow students will now present some basic facts about tuberculosis and its testing, explain how our biosensor should be constructed, and why we believe it has the potential to change modern latent tuberculosis infection testing.
Tuberculosis is an infectious lung disease caused by Mycobacterium tuberculosis. About 2 billion people are now infected with TB. However, 90 to 95% have a latent form of TB, meaning they have no symptoms. However, it can reactivate when the immune system is weakened, causing infection. If not treated, the infection can spread to other tissues in the body, affecting organs like kidneys, brain, or liver. TB caused more deaths than any other infectious disease. Epidemic of tuberculosis caused about 30% deaths in Europe in 17th century. By the late 19th century, 90% of urban population were infected with TB, and about 80% of those who developed the active form died. TB is also commonly called white death due to the extreme anemic pallor of those infected and a robber of youth, because the disease had higher mortality rates among young people. Up to this day, TB remains a significant challenge. In 1993, World Health Organization declared tuberculosis a global emergency. It's mainly treated with antibiotics like isoniazid and rifampicin. However, those drugs are ineffective to multi-drug-resistant TB. Second-line drugs are required, which are mainly too expensive for developing countries to treat it successfully. It is predicted that from 2 to 3 million people will die from TB every year, which is approximately 1 death every 10 seconds.
Before we jump to explaining how our biosensor will be built and how it will work, we need to address the role of the key molecule involved in the fight against the infection: interferon gamma. I believe that most people encounter the term interferon for the first time in the context of fighting viral infections in the body. And while it is partially true, this group of molecules play a pivotal role also in other immunological pathways. In general terms, interferons are cytokines, proteins with signaling functions. They can be divided into 3 types, out of which type 1 and type 3, although secreted by different types of cells, are said to be mainly involved in limiting the spread of viral infections. Our biosensor will be designed to recognize the only member of the type 2 interferon group, interferon gamma. Its main role is to activate macrophages and support cell-mediated immunity in general. It's normally secreted by T cells, but there are other types of immune cells that are able to produce it, for example natural killer cells and macrophages. So, how is interferon gamma involved in fighting off Mycobacterium tuberculosis? First, parts of the pathogen are presented by the antigen-presenting cells, which also start producing interleukins, other signaling proteins. This leads to the activation of the cells, which are able to produce interferon gamma. Secretion of this protein changes the activity of other immune cells. For example, it can result in the change of the class of the antibodies produced by B cells, or stimulation of macrophages, or it can further enhance the pathways of processing and presenting the antigens, boosting the immune response. The most important thing is that levels of interferon gamma increase during tuberculosis, which is why many diagnostic tests rely on measuring them.
Correct and efficient diagnosis of infection is crucial in fighting tuberculosis. Right now, we have several strategies in detecting ongoing infection. To detect an active form, we mostly use rapid molecular tuberculosis tests that target particular sequence of nucleic acids. This also allows us to identify antibiotic resistance of the infecting strain by using sequencing or nucleic acid hybridization. In the case of latent tuberculosis infection, in short LTBI, the standard evidence of tuberculosis isn't always manifested, even though the organism is infected. In this situation, World Health Organization recommends using skin test or blood test. The first one is antigen-based tuberculin skin test. It is performed by injecting tuberculin into the skin on forearm. Then, after 2-3 days, the doctor diagnoses the patient based on the appearance of the injection spot. The blood test is interferon gamma release assays. It is based on laboratory analysis of the patient's blood using antigens specific to Mycobacterium tuberculosis. Both methods are useful but still have limitations, especially when it comes to detecting LTBI. And that is why we want to propose the use of biosensor of interferon gamma based on Escherichia coli. That may be a method potentially allowing for the diagnosis of latent tuberculosis infection.
Before delving into the details of our project, let's take a closer look at biosensors in general. They are analytical devices that make use of biological components, and their goal is to detect particles or phenomena. The biological origin of said components significantly influences the properties of these systems, as it guarantees high sensitivity as well as specificity, while also reducing the costs of the production of the sensors. Biosensors consist of 3 basic elements. The first one being the bioreceptor, which is responsible for detecting specific compounds or physical phenomena. That detection in turn generates a signal that is received by the second element of the biosensor, that is the transducer. It also converts the signal into, for example, light, which is information that is easier to read by the third element, which is the detector. The detector registers the signal and converts it again into the information that is most useful to the researcher. Biosensors can be based on whole cells, and one of the microorganisms that is most commonly used to construct such biosensors is Escherichia coli. The biggest advantages of using these bacteria include the fact that its structure, physiology, and genome are well known. It is also characterized by fast growth, on-chip media, and there are non-pathogenic laboratory strains available. Moving on to the human interferon gamma biosensor. It is based on Escherichia coli cells and has 2 key components. First one being interferon gamma detection and second one being fluorescence generation. Interferon gamma cannot penetrate the outer membrane, and so the sensor needs to be able to detect interferon gamma from the outside of the cell. To do that, the chimeric protein called IFN-γ is used. It consists of parts of 2 orthologs, first one being OMPA, which is a transmembrane protein very common in gram-negative bacteria, and it provides a beta barrel that anchors the chimeric protein in the outer membrane. However, particular extracellular loops are replaced with those deriving from the second ortholog, and that is OPRF. Its function is to recognize and bind human interferon gamma. Seeing as efficient expression of the sensing component is crucial for the functioning of the whole biosensor, IFN-γ coding sequence is fused with a strong promoter as well as RBS originating from the tetracycline resistance gene. The signal generated by IFN-γ binding is transduced by conserved in Escherichia coli phage shock protein system. As a result, PSPF transcription factor is released and able to activate transcription from PSPA promoter. In the interferon gamma sensing system, a reporter gene is expressed from that promoter instead of a phage shock operon. In our case, that reporter is blue fluorescence protein. That allows for obtaining signal that is easy to read and gives us direct information about the presence or absence of human interferon gamma particles.
After presenting the theoretical aspects, I would like to talk about plant laboratory procedures. The standard technique which will be used in the wet lab with the 3A assembly very often used in synthetic biology. This assembly allows for quick ligation of biobrick flanked by prefix and suffix substrates, which came from different plasmids into one vector. The use of 3 different resistance ensured 97% confidence that only the correct colonies will be obtained after transformation. In this slide you can see the structure of our biosensor. The first part is a receptor, which contains a system where the chimeric IFN protein is under the control of tetracycline promoter. The gene encoding the protein is followed by a double terminator sequence according to the RFC10 standard. Signal transduction is mediated by PSP proteins that induce the expression of genes under the control of PSPA promoter. In our case, it will be the BFP reporter gene. Our work will be concentrated on 3 steps: assembly of the reporter expression system, assembly of the receptor expression system, and testing the functionality of the biosensor. As a result, we aim to gain a functional gamma interferon biosensor. We will start the assembly of the reporter by connecting the gene encoding BFP with the terminator according to the 3A assembly protocol using iGEM assembly kit. The product of the reaction will be transformed into chemical competent bacteria, which then be sown on an LB medium supplemented with antibiotic according to the backbone resistance. Some grown colonies will be relatively sieved onto fresh LB medium to confirm a positive transformation results and to obtain a pure culture from which the plasmid will be isolated with the commercially available kit. The following promoters will then be added to the BFP terminator system. Lactose promoter, which will be a PCR reaction product on E. coli top 10 strain using specific primers with overhanging suffix and prefix. Tetracycline promoter, which will also be a PCR reaction product on isolated PSB1 AK3 plasmids using specific primers with overhanging suffix and prefix. PJ23 isolated from the plasmid by cutting the suffix and prefix sequences with appropriate enzymes, and PSPA cloned in the same way. All 4 promoters will be first ligated with biobrick RBS, and after transformation, grown and plasmid isolation will be used to ligation with BFP terminator system. 4 mixtures of ligation reaction will be then used to transform bacteria, which after being brought through reactive sieving will be the material for assessing the strength of PSPA and tetracycline promoters by measuring the fluorescence of BFP after induction. PJ23 promoter as a constitutive one will not be inducted. Measurements will be made on 96 well plate using a reader enabling the excitation of BFP and registration of emission waves. 3 technical repetitions will be used to calculate the average of measurements, which will be then normalized by culture optical density. We assumed that PSPA promoter activity will provide a high level of reporter, what will make the BFP level in functionating biosensor easy to detect. Also, a tetracycline promoter is expected to be strong in this system, which will provide a proper amount of receptor part, determining the biosensor sensitivity. The next step will be to add the IFN double terminator constructed at the Department of Bacterial Physiology of our faculty to create an earlier tetracycline promoter with RBS. The complete part will be also transformed into E. coli bacteria, which will then be brought through reactively sieving. The last step of construction will be to combine both parts into one vector, also using the 3A technique. After transformation and bacterial growth, the culture will be the material for research on the functionality of the biosensor after prior sequencing of the entire biological part in order to confirm the absence of sequences error. A biosensor activity test will be done with bacterial culture diluted to the optical density of 0.05 with fresh LB medium and incubated to 0.2 prior the addition of an inductor. The active response range to gamma interferon will be examined at concentration of 0 to 1000 picomolai. After incubation with an inductor, we will measure the fluorescence as it was made before using 96 well plate. Average of measurement will be normalized by optical density and reported as RFE per optical density. Testing of the biosensor system will take place at inductor concentration in the range of 0 to 1000 picomolai to obtain information about wide range of biosensor operation. Based on available data, we predict a significant increase in fluorescence level beyond the lowest tested values and a slowly increasing level in a higher inductor concentration. If the expected results coincide with actual ones, we plan to test the biosensor also at higher inductor concentration to test the receptor saturation and to perform tests on material from living donors. Also, if laboratory work will run without major problems, we plan to optimize the operation of the biosensor, in which, among other things, various RBS systems will be tested.
The constructed biosensor will have to be tested and optimized using interferon gamma protein, preferably humankind. Working with factors that regulate the human immune system always generates risks. For example, bacteria developing new methods of responding to said factors. Therefore, all wet lab experiments should be performed in the spirit of the biorisk management culture and by researchers who underwent safety training. Another possible solution to this issue is using mice's interferon gamma, but this may cause optimization to be less effective. Another potential problem is that the OPRF protein is a virulence factor in Pseudomonas aeruginosa. Nevertheless, only small fragments of the OPRF responsible for binding interferon gamma are used to assemble IFNs. Proper isolation of those fragments, without transferring Pseudomonas aeruginosa cells, should resolve this issue altogether. There is one more problem concerning the usage of the biosensor. Since the proposed device will be used among low-income populations, the risk of environmental contamination with modified Escherichia coli cells is relatively high when the biosensor is not appropriately handled. This being the case, tests should be performed by trained medical practitioners who understand the risks of leaking bacterial cells to the environment. To sum up, even though there are many promising theoretical indications that the proposed biosensor could become a satisfactory alternative for standard latent tuberculosis infection testing, experimental studies are vital to acquire a definitive answer. Naturally, the biosensor constructed in the described way should be utterly tested and optimized to evaluate its sensitivity, reliability, and therefore its practicality. Of course, actually preparing and implementing this technology will be a long and challenging but rewarding process that may contribute to the quality of latent tuberculosis infection testing. Here you can see a list of publications that we used while preparing this proposal. And thank you very much for your attention. We hope that our presentation caught your interest.